Hi, my name is Marlaina Horewitz, and this is a presentation about the paper I wrote this summer about arginine-rich HTPs. I decided to study them due to some very odd results I received while performing circulated coism experiments on TP4 as a member of the Cotten lab in the Applied Science Department at William & Mary. Host defense peptides are used by the immune system of an organism to identify, deliver drugs to, and sometimes neutralize threats to the organism. These threats can be tumor, bacteria, or even viruses. Some HTPs are cell-penetrating peptides. These are peptides that can get past the semi-permeable lipid bilayer, which makes up the cell membrane of various types of cells. Some CPPs fully internalize into the cell, while others only partially insert themselves and remain in the cell membrane. They usually contain a lot of arginine. However, the exact method of insertion and the method of neutralization of the threat is highly dependent on the peptide structure. That is why in the Cotten lab, which studies these cell-penetrating host defense peptides, we are often focused on the structure of the peptide at various points in time as it relates to functioning. Tilapia piscidin 4 is a host defense peptide found in fish. Notice in the sequence the +5 charge in the end caused by the amino acid arginine, abbreviated R. When using artificial mimics in the lab, as well as in some in vivo experiments, it has been shown to have some cancer and infection-fighting abilities. Specifically, TP4 has been found to be effective at neutralizing an antibiotic-resistant bacteria called Clostridium difficile. That's why it's such of an interest to the Cotten lab. It has the potential to be a treatment for an infection for which there is not really a great treatment out there. However, to utilize it as a treatment, it must not affect normal cells significantly. There's been some promising experimental data to support this claim of specificity. My goal in completing and writing a literature review is to formulate a testable hypothesis about TP4 and how it inserts into the membrane and kills the cells. To capitalize on the promising possibilities for cancer or infection treatments, we must first understand how the peptide is able to perform its functions in order to minimize side effects and to be able to create an informed cost-benefit analysis of utilizing these peptides. That hypothesis is quite complicated, so in the paper I wrote, I decided to break it down into 4 main parts. The first is specificity due to the positive charge. According to Coulomb's law, negative charges are attracted to positive charges. So considering that arginine-rich HTPs are positively charged and bacterial and tumor cells have a negative charge, it is logical that this may be linked to their specificity. There's further evidence to support this. When these peptides were in an environment with both neutrally charged cell membrane mimics and negatively charged cell membrane mimics, they insert into the negatively charged cell membrane at a much higher rate than the neutrally charged membranes. Hydrogen bonding is a great explanation for the energetic cost of insertion. Since peptides insert without a vesicle, they need a source of energy, and arginine-rich peptides can make up to 6 hydrogen bonds per arginine to provide such energy. In my hypothesis, the method of cell death is apoptosis, which is programmed cell death. This is supported by one key piece of evidence. Capsase-3 is a signaling molecule which has been shown to lead to apoptosis. It has also been shown that when live cells were exposed to arginine-rich peptides and subsequently died, capsase-3 was present in high concentration. Finally, I hypothesized that this apoptosis was triggered by a change in voltage potential. To make ATP, the form of energy used in our cells, ATC synthase relies on a concentration gradient to spin. The presence of a positive charge from arginine can disrupt this gradient and cause the cell to starve and subsequently induce apoptosis. Energetically and functionally, this does make sense. I will do assays with live cells to verify this hypothesis. Here are my sources. Thank you for listening.